All right. Thanks, everyone. Thank you for the introduction and the kind invitation to present my work. So yeah, today I'm gonna talk about models, stochastic models of division control. So models we make to understand better this fundamental question. So whether cells, how do cells know when it's the right time to divide? So in a typical experiments, we collect a movie like the one I'm showing you, in which cells are growing in a given device under a microscope, and they are growing and dividing many times. And we want to know basically how whether this decision to divide is taken completely at random or in a more, let's say, meaningful way. And this question is motivated by two key observations that you can make by studying this kind of data, this kind of movies. The first one is that if you look at the cell size at the birth size, so at the beginning of each new cell cycle, which is the period of time between birth and division, this process is stable. So cells tends to maintain a stable cell size as they continue to grow and divide over time. Not only that, but they also control their distribution of cell size, which is narrowly distributed with typical fluctuation of about 10 to 20% around the mean. And this is thought to be essential for their behavior, basically. So again, we want to understand how these cells take this decision better, basically, with this kind of model that I'm going to present. And of course, you can take two different approaches depending on the level of details you want to include in your model. Today, I'm going to discuss two distinct approaches, one more phenomenological and one more molecular. So basically, you can consider cell basically as a container of a given size, growing for a given division time at a given growth rate. And so in this, let's say, phenomenological approach, you would study correlation between quantities such like birth size, division time, growth rate. Or you can, of course, view as the cell as a more detailed object made up of proteins that might be essential for the division process itself, but also made up of chromosome, ribosome. So you want to understand how the division process emerge from the dynamics of these ingredients. And of course, the hope will be in the end to kind of connect these two level of details. So you want to somehow being able to reproduce the correlation pattern that you can see in your data at a phenomenological level by means of a more molecular description of the behavior. So I'm going to start with the phenomenological theory, which will be helpful also to set a lot of notation and key ideas. So basically, the key observables we will look at are, again, the cell size at birth or at division, S0I and SFI at a given cell cycle I. And other key quantities are the growth rate, so the speed of growth, how fast cells are growing, alpha, and the division time, so the time it takes for a cell to divide from birth. And then I'm introducing also two other key quantities, QI, which is the logarithmic size, and the GI, which is the overall added logarithmic size in a given cell cycle I. So within this phenomenological framework, we can immediately realize that the question, at least for exponentially growing cells, is not a trivial question, because one immediately realizes that as soon as fluctuations are allowed in the either growth rate or division time, these fluctuations, if are just random noise, they will add up to make this trajectory diverge. And we can see this, for example, writing the exponential law of growth, stating that the final size is the initial size times an exponential. And we can take the logarithm to write basically an autoregressive process for the logarithmic burst size, relating the fluctuation in the logarithmic size at the next cycle to the fluctuation in the logarithmic size at the current cycle plus a fluctuation in the added logarithmic size, which can result from either fluctuation in either the growth rate or the doubling time. And so we can see that if this term is just a random noise, this process is just describing a random walk, which we know is not stable at all. So this means that if these fluctuations are just uncoupled, are just random, we will see in our experiments single trajectories diverging over time, which is not, of course, what we see in a typical experiments. So one immediately realizes that this fluctuation must be somehow coupled to fluctuation in the initial size, meaning that cells must somehow know how bigger or smaller than average they are at the beginning of a given cell cycle, and somehow tune either the growth rate or the division time to compensate for this fluctuation. For example, cells that are born bigger than average, they can decide to spend less time in a given cell cycle so as to compensate for fluctuation. And we can express this fact by just saying mathematically that the fluctuation in the added logarithmic size, GI, should be a given function of the fluctuation of the logarithmic burst size and noise. So this would be the starting point of the, let's say, a phenomenological approach to the cell division control problem that we are going to address now. So starting from this expression, of course, this function we expect being very highly nonlinear, highly complex. We don't know actually the exact form of this function. So to progress with this theory, we need to make some assumptions. And usually one assumes, for example, that the noise term is an additive noise term, and this is motivated by experiments. And this noise term might result, for example, for noise in either the growth rate or the doubling time. And then one approximate Taylor expand to first order this control function, basically recapitulating all its complexity within a given single control parameter lambda g. And again, this approximation is justified by the fact that I told you that the distribution of this fluctuation is typically very narrow. So one might hope that truncating the Taylor expansion to first order might be enough to, let's say, recapitulate all the complexity of the process here. And so basically we end up with the first order autoregressive process linking the fluctuation in the burst size at the next cycle to the one at the current cycle plus a noise term. And here we have a multiplicative factor, 1 minus lambda g, that will tell us the strength of the control. And of course, by just iterating this equation, one can see that on the long run, that is for I way greater than 1, this process is stable, will lead to a stationary size distribution, as provided 1 minus lambda g is in modulus less than 1. And in particular, for lambda g positive, this can also be viewed as a discrete time or Stein-Uhlenbeck process with exponentially decaying autocorrelation. So you can see that we have a finite expression for the variance of the logarithmic burst size, which can be written in terms of the variance in the additive noise. So this might be a timing noise, for example, divided by a correction term that depends on the actual division strategy implemented by the cells. And then we can also write all the different correlation across subsequent cycles, for example. Okay, so this lambda g then is a crucial parameter in this kind of models. And of course, it can assume any values between 0 and 1. But typically in this theory, three prototypical values are considered because they have clear interpretation, which are 0, 1/2, and 1, and are respectively called timer, adder, and sizer division strategies. So for example, for a timer, lambda g equal to 0, this corresponds to a strategy in which division occurs after a constant time, or a constant logarithmic growth. Because you can see that the final size would just be twice the initial size, irrespectively of any of the initial size, basically. So if growth rate is constant, this is called a timer because this is equivalent to dividing after a constant time. So basically the time you need to duplicate your initial size. And as we already seen, this is the strategies that leads to a random walk. So here fluctuations are not controlled, basically. And indeed, you see that the fluctuation in this initial size at the next cycle is just the fluctuation you have at the current cycle. So you are not controlling fluctuation, basically. So you can have an intermediate control called adder. And this is called adder because you can see that according to this quantity, the final size can be written as the initial size plus a constant size-independent term plus some additive noise. And so this means that you divide according to a strategies, so that you divide after adding a constant size, basically, independently of your initial size. I mean, the other size is independent of the initial size. And here you can see that you are controlling size fluctuation, but you are controlling it, you are losing memory basically exponentially in time, as in a Orstein-Uhlenbeck process. And then you have the strongest strategies, which is called the sizer, which basically corresponds to a value of lambda g equals to 1, which immediately in just one cycle control the fluctuation in the initial size. So basically, according to this strategy, you divide at a given target size at any cycle. So one might, you can see that any different value of lambda g, so for example, these three values I was discussing, predicts very different, let's say, correlation between the final size and the initial size. So one can turn to data, then compute this correlation and see whether nature favor one of the three strategies, for example. So this is the kind of analysis we can do within this phenomenological theory. And if you do this, you see that you see a sort of universality of the adder control, despite I told you that the adder strategy is a sort of intermediate strategy. So you might think that cells want to kind of control immediately within just one cycle of the fluctuation they have, if the aim is to stay within a given target size, but this is not what is seen in nature. And this is some of the questions that one might want to understand better with a more microscopic theory, and we see a possible answer to a possible explanation to this phenomenon. And also, this linearized phenomenological framework, why it's very powerful to study cell division control and also to select different models with data. It also lacks any connection, of course, with the underlying molecular mechanism. So, for example, we don't know how the other strategies could emerge from an underlying molecular process. And also what are the effects of distinct mechanistic ingredients insecting the division strategy or size fluctuation, for example. And so these are all kind of questions that we would like to understand better with a more mechanistic and microscopic description. So turning to this more mechanistic approach, again, as I told you, the aim here would be to describe the dynamics of some key molecular player driving the division process. For example, some key players might be some key divisor protein, but also the chromosome might play a role, as well as the ribosome is synthesizing new proteins. So one might want to define a dynamical equation for these ingredients and then connect these their dynamics with the phenomenological pattern that we just discussed. So I'm going to present so while the phenomenological process been largely investigated in literature, these kind of molecular approaches are less investigated both theoretically and experimentally. And I'm going to present some recent results, some attempts to do that with just one divisor protein. So basically, all the details inside the cells will be neglected apart from one key molecule X, which is thought to be limiting for the cell division. And of course, we need to then specify the law, how these proteins accumulates, and how the decision of dividing is taken depending on the dynamics of the protein. So these will be the two ingredients that define our model, which is called threshold accumulation model. And basically, according to this model, you are synthesizing this divisor molecule X at a rate that is proportional to size. So the bigger you are, the faster you are synthesizing this protein. And so you are making a new protein and a new synthesis event with this rate. And the logic for cell division is very simple. So basically, you divide once you have enough of these proteins. So you have a threshold, which for the moment is fixed. And once you hit the threshold, you divide. So you see here, for example, an example of a simulation of this process. So you have a cell that is increasing size deterministically. And on top of that, you have the accumulation of this molecule. And once the molecule hits a threshold, it divides and the process starts again. It divides and the process starts again, and so on. And so, of course, you can describe the dynamics of this, the accumulation of these molecules as a non-homogeneous Poisson process with the following master equation. But more importantly, in this kind of model, division timing becomes the first passage time of a non-homogeneous Poisson process to this threshold. And what's interesting is that from the statistics of the first passage time, we can retrieve our phenomenological quantities, lambda g, which I remember you that quantifies the strength of division control, and the coefficient of variation of size fluctuation, basically. And so basically these equations are just another way of defining lambda g. I told you that lambda g was defined as the derivative of the control function, which is basically the added size. And so the control function could be thought as the conditional average of the interdivision time conditional of the initial size at a given cell cycle. So this is just another way of stating the definition of this parameter. But importantly, in this way, we can connect directly its definition with the statistics that we can measure for this more like molecular model of division control. Okay, so the conditional average, it's no result of non-homogeneous Poisson process that can be written in this term, in this way, where this variable y is gamma distributed. So then exploiting Gaussian approximation for this gamma distribution, justified by the fact that the threshold is typical high, we can compute the derivative and compute the lambda g. And we see that lambda g is equal to 1/2, independently of any model parameters like theta or r, which is the reset fraction, or the synthesis rates of the molecule. So basically, this model is intrinsically implementing another strategy. And you see that here you see the comparison of this prediction with simulation of the model, varying different parameters, for example, the molecular threshold. So this is a simple way then to implement another at a more, let's say, a mechanistic level of description. So how are fluctuation in this model? So if you compute the CV square with the recipe that I told you before, you see that as expected, you have a Poisson scaling of size fluctuations, meaning that the CV has a 1 over square root of theta, where theta is the threshold, scaling of size fluctuation. And so basically we can then see what is the amount of proteins you need to match the fluctuation you predict with this kind of model with the empirical ones, which I remember you that are around 10 to 20%. So you need, oh, sorry, I zoom in. Okay, so you need around 20 to 30 molecules to to produce enough the empirically observed fluctuation. And so one can see then what are the typical values of these divisor molecules. And if you look in the literature, in the experiment, the typical copy number of these plausible divisor proteins are more around 1,000. So this means that while this model implements an adder, which is okay for us, since adder is widespread, it cannot alone explain all the entire variability of size that you see in a typical experiment. So this model should be refined in such a way to increase the size variability, in other words. And a way to do that, which was explored in a recent paper, was to add some ingredients to the synthesis dynamics of the protein. So basically, you can add a constant production term, nu, in the synthesis dynamics of the protein, or a degradation term in the protein. So now you no longer have just production, but you have also loss of this molecule. So this is now, more generally, is now a birth-death process. So we are in a different class of stochastic process, but you can then work out the model, the main quantities in the same way, starting from the master equation, for example. And you see that the addition of these two quantities indeed increase size fluctuation from the intrinsic behavior. So for example, here you see that by increasing the production, the constant production rate, you increase size fluctuation. And this is true also if you increase degradation. So basically, because you are basically allowing for more events, accumulation events, to get the threshold. So all the dynamics will become more noisy. But you also have the side effect of changing of the division control strategy. So if you increase constant production, you are shifting division control toward more timer-like strategies. So strategies appear tends to divide after waiting a constant time, whereas if you increase degradation term, you are shifting your division control strategies toward more sizer-like strategy. So here you are more dividing after reaching a constant size. So you see that you have this kind of trade-off. And interesting in this paper, they also saw that the other strategy becomes the ones that minimize size fluctuations. So you see here in this plot, for example, where they plot the rescaled coefficient of variation versus this parameter lambda g. And you see that you have a minimum in correspondence of the other. So this might answer our initial question of why cells should implement a sort of intermediate strategy in terms of controlling size fluctuation. This is because if you consider molecular noise, this intermediate strategy is the one that is minimizing the overall size noise. And this is because if you look at the phenomenological CV square equation that we wrote initially, so I told you that there was a contribution from the fluctuation in the timing noise and a contribution from the division control. But actually what they figured out, which is very interesting to me, is that also the term in the numerator has a lambda g dependence. So it's not if this wasn't the case, of course this term would be minimal at lambda g equal to 1, which is the sizer. But actually, since molecular noise makes this quantity depending on lambda g, they found that the actual minimum is in correspondence to the other. And this is due to a trade-off between minimizing timing noise and minimizing this term, which is called homeostasis factor in their paper, basically. However, we recently found, and all this was obtained in this model where you just consider molecular noise. However, we found that this optimal strategy is another only in this intrinsic noise limit. Whereas as soon as you include other sources of noise, these minima shift toward more Sazer-like, but it can shift also toward more timer-like behavior. So more in general, optimal control, if exists, is actually noise dependent in a more general framework in which you also consider extrinsic noise sources. And what is this framework? So we consider basically the initial simple accumulation dynamics for the molecular divisor molecule. So you synthesize this molecule with just a size proportional term. But then on top of it, on top of that, you consider fluctuation, extrinsic noise, and basically we consider fluctuations in the threshold. And we model fluctuation in the threshold as a Orstein-Uhlenbeck process. So you have a given memory raw with the previous fluctuation and a sort of independent noise term eta I. So you see that you have basically two dimensionless parameters describing the different regimes in which you can operate. You have this parameter rho that quantifies the strength of memory between differences in threshold fluctuations between different cycles. And you have this sigma, big sigma, that quantifies the strength of extrinsic noise, so noise in the threshold level versus the molecular noise, so the intrinsic noise we just discussed. And you see that you can go from a purely uncorrelated noisy threshold, where you have a fluctuating threshold, which is just uncorrelated within selves, or toward a pure correlated noisy threshold. Okay. And of course, you can write the master equation, solve the first passage time, and compute the division control and size fluctuation variables. And what is interesting is that if you do the math, you can write these two quantities as their intrinsic values that we just discussed, times a modulation that depends on these two quantities that we introduced. So the memory between different cell cycles and the strength of threshold fluctuation. And so the key result, and you see in the right plot that you can, for example, for lambda g, depending on the value of the threshold memory and this extrinsic to intrinsic noise ratio, you can make this model behave as you want, basically. So you can have either adder, sizer, or timer, or anything in between. And of course, you have different limits in which you have different division control strategies. And also, importantly, you can go from negligible to experimentally relevant fluctuations in the cell size. And the reason why you can tune these two parameters is that adding extrinsic noise, you create a sort of memory between subsequent cell cycles. So, for example, you see that the fluctuation in the added amount of molecule that you need to synthesize, which is proportional to the fluctuation of the added size in this kind of model, will be dependent to threshold fluctuation in the preceding cell cycle. So you are kind of introduce a sort of memory, sort of correlation between the added size and the previous cell cycle, which is allowing you to tune the division control, basically. And so this sort of memory you are introducing is dependent of the difference between raw, which is the memory between threshold levels, and this R parameter, which is my reset. So basically, this R parameter is telling me what is the fraction of protein in the threshold that I maintain at the next cell cycle. So both quantities introduce a sort of memory in the system, and depending on the balance between these two quantities, you can either go toward adder-like, toward sizer-like or timer-like division strategies. But interestingly, there is a regime in which you lose this memory For example, when rho is equal to r.
Mattia, you should come to an end.
I'm basically done. Okay. So basically, we just to conclude, we found a regime in which when rho is equal to 1, you have other control, but you can independently tune self-fluctuation. So basically, you are solving the problem we had at the beginning because we can have an adder, basically, and we can tune size fluctuations so as to fit the data. And so by comparing the data with the prediction of the model, we saw that you need actually these sources of these extrinsic noise sources to capture the variability you see in E. coli data. And so with that, I'm concluding. The take-home messages are that there is a universal adder control and tight size fluctuation in different organisms. I show to you a simple model, mechanistic model, in which you can have other control, although other mechanisms such as DNA replication surely play a role. If you just consider molecular fluctuation, you can explain the adder, but you cannot explain the actual size variability you see in the data. And basically, we found a way to reconcile both quantities, basically. So with that, I would like to thank you for the attention and all the collaborators in this project, which is now out on arXiv. So if you want to check out more details, you can do basically that. And I thank you again for listening.
Okay, thank you, Mattia. So we have time for just one or two short questions, if any.
Perhaps I have one, Federico.
Stefano.
Just a curiosity, would it be a reason to assume that this lambda g in your first model is itself a stochastic variable? Because this will change a lot the statistical properties of the process. It will become a multiplicative noise process, and it will change a lot the final statistics, right?
It might be reasonable to assume that lambda g is not just fixed at all cell cycles, but 2 as a cell cycle dependency. So you will write lambda g I. So at each cell cycle, cell would implement a fluctuating control strategies, and then you could write a stochastic dynamics for lambda g as well, I guess. So, but yeah, usually, there is a work where they look at the dynamics of lambda g during also a shift or in stationary condition, experimental condition, and they saw that it's pretty stable. But in principle, you can.
It's not motivated by observation in a sense.
Yeah.
And another quick question. The fact that you see an optimum in this process, no, an optimal lambda g that seems to give you the least variability. Could it be connected with some form of entropy minimization in a sense? Because in a sense, it's like a condition in which you want to make the number of states produced per unit time. I don't know. It's very
I think
vague question.
I think you can frame all these framework as an information processing, let's say, task in a way, and in that way you could find a matching between this minimization to a more like physical or entropy related minimization. I don't know of work that have followed that line, but I guess it might be done.
Okay. Thank you very much.
Thanks.
Okay, so let's thank Mattia again.